This video is about DNA, deoxyribose nucleic acid. So to begin with, DNA is made of a backbone of deoxyribose and phosphate and deoxyribose and phosphate, etc. This squiggly line just means it carries on going. Now each deoxyribose is linked to a base, more on the base later, and there's a hydrogen bond that exists between the bases of the two DNA strands, and it's in a double helix. So before we get to the details of the DNA structure, we're going to just break it down a little bit. We're going to start off by looking at the phosphate. So the phosphate, based on phosphoric acid, and it has a so phosphoric acid has additional H here, so it's H3PO4. But the tendency is the phosphate group loses all of its hydrogens. It depends on the scenario that if we're making a nucleotide, we'll see later, you may see an H left, but this is the phosphate group as seen in the data sheet. So the O- exists, and then the H, two H's on the other end, they will drop off, so to speak, when they form the backbone of the DNA. And so this is how it exists, where this just means the DNA is continuing. The 2-dehydroxyribose contains 5 carbons, and it's very important that you appreciate that knowing the positions of these carbons is essential, so 1 to 5, and they tend to have these in the textbooks, they're numbered 1 to 5 in this way, and I'll refer to those throughout. Now 2-deoxyribose comes from the fact that this group here, this 2 carbon, has lost an oxygen, hence deoxy. So there's ribose here, C5H10O5, loses the oxygen, making C5H10O4, and this 2-position oxygen has been lost from this carbon, hence 2-deoxyribose. So a bit more about the backbone of the DNA. So just put a little bond in there, I think. There's a bond there missing. So we got the backbone of the DNA again, phosphate, this is what we're going to focus on, phosphate deoxyribose, phosphate deoxyribose, continuing. And what happens is there are connections between the oxygen on carbon 5 to the phosphorus, and the oxygen will connect to another carbon of another deoxyribose. Carbon 3 here reacts with another phosphate group, and it's the oxygen which links with the phosphorus here. And now we see the OCH2 again linking to the phosphorus as we did here. So that continues. So it's always a P bond to an O here next to the 5, and the O of carbon 3 links to the phosphorus at the other end of the deoxyribose molecule. So it's 3 and 5 which are important there. So now there are no H's shown on the phosphate groups. They've been lost to become water. It's a condensation reaction, more on that later. This is not a full picture of the DNA because we haven't added any base pairs yet, which will link to these carbons here. Sorry, these ones here. The OH here will fall off when we form DNA. So the base pair is attached to carbon 1 by a CN bond. So again, back to 2-deoxyribose, where we see that this carbon group here will be essential for connecting to the base pair, or the base that will form the base pair with another DNA strand. And then these are the bases here, adenine, thymine, cytosine and guanine. And this group here, the circled ones, always connects to carbon 1. And so what happens is the OH here and the H of the base form water, and this N, the ones I've just dotted there, they form a bond with the carbon number 1 on the deoxyribose. I just state a little bit more about the fact there's a 6 and a 5 carbon atom ring, 6 for thymine, cytosine has got 6, and guanine is made of these two cyclic areas here of within the structure. So all of these base pairs can link to carbon 1. Sorry, bases can link to carbon there. So key term is nucleotide. Nucleotide is a phosphate, deoxyribose, and a base. Not a base pair, a base. So we have a, this is how it works. So you've seen this earlier on with the carbon 1 linking to the phosphate group there. Notice how that H hasn't been lost yet, but it could very easily be lost when it reacts with another deoxyribose up there. Then we have the another group, another deoxyribose phosphate group can be down here. And what I've showing here is the carbon number 1 is now linked to the base. This base is adenine. It has this name which you don't need to be aware of, but it's adenosine monophosphate. So that's a nucleotide. That whole part there is a nucleotide. I'm just showing here the way that the hydrogen bonds can form with another base. With guanine, there's a similar picture. Again, carbon 1 has now got the base attached. 3 hydrogen bonds form from the guanine. With a cytosine, we see that the carbon 1 has got the base there with the 3 hydrogen bonds. So it's the CNG which will link together when they form the base pair of the two double helix. And here we have carbon 1, thymine this time, linked to the carbon there. So all very similar. So these are a single nucleotide with a phosphate group, deoxyribose group, and a base group. So you might be asked to draw the structures, but remember that you know structures like this, where you're phosphate, deoxyribose, and base, but you have the different structures given to you. These little segments will be given to you on the data sheet, so you can look those up. But what you're not told is that the base connects to 1 and the phosphate group connects to the carbon group here. So DNA structure formation. This slide really is just talking about the importance of condensation. So first of all, forming DNA, the phosphoric acid group or the phosphate group will react with the carbon group here, and it's a nucleophilic attack really. This oxygen will attack the phosphorus there, but making water as a byproduct. So this OH and this H make the water, and now we have this new bond between the oxygen there and the phosphorus there. So that's the deoxyribose and the phosphate group with water as a byproduct. This could also react in a similar way with another phosphate available there. That reaction will also occur, also condensation, polymerization. And so the next part of the DNA strand is to make this this bond here between the carbon number 1 and the nitrogen group. So again, I'm highlighting the H here, which will be lost, and the OH here lost. This is the nucleophile in this case. Water is the byproduct. And so we now have this carbon group here. We are missing a double bondo there. So the double bondo there, and also this can also react with another nucleotide. So this has the ability to react with another nucleotide this end to make a long strand of DNA, and this can make another react with another nucleotide there, and overall making DNA. And the last part of this video is talking through how you should be able to show how the base pairs interact. So the base is always in the same pairs. So A and T and G and C. Between A and T, you've got 2 hydrogen bonds and 3 hydrogen bonds between G and C. Now, this is how the structures are printed on your data sheet. So what I suggest you do is get into the habit of, when you see a data sheet, think, well, these are the sort of the top left of the adenine. These are the atoms which form the hydrogen bond with the thymine. But you've got to understand how it works, and we'll see how it works in a sec. It's just hydrogen bonding between H and O molecules, or atoms on the molecules, and this is the structure that will disconnect. So I would suggest circling the atoms on your data sheet, so you know that these are hydrogen bonds for cytosine and these are the NH. This NH will react with the carbon 1 of the deoxyribose, and all the cases here. So circle these ideas, these will connect these, structures will connect with these structures here will connect with those ones there through hydrogen bonding. Remember, these are how the structures are drawn on the data sheet. And what you may be able to do, is to draw the structures between base pairs. So between thymine and adenine, we get the H here showing the hydrogen bond to the oxygen, and the H on this thymine structure bonding to the N of the adenine. Cytosine, O to H, H to N, and O to H. This is a very simplistic diagram of the hydrogen bonding. Really, we should show delta pluses, delta minuses, all lone pairs, but I've simplified it here. So this is how we can show hydrogen bonds between base pairs. They may draw this structure, and then you're asked to select the correct base pair and then connect it with the thymine.